Services and Outreach at the University of Chicago for the GDC project, and I have with me my colleague Colin Reed. And today we're going to talk a little bit about some of the data analysis, visualization, and exploration tools in the GDC. Actually, before we get started, we're going to ask a couple quick questions. Well, first, some bookkeeping, in that you're all going to be muted by default, just to preserve the audio quality. So please type your questions that you may have into the chat screen, and we're going to reserve a bunch of time at the end so we can answer all of those questions. The other thing that we wanted to do at the beginning is, if you wouldn't mind, totally voluntarily, just write in the chat screen the organization from. It's just a curiosity of us to see, who we're reaching. It's up to you, but we'd appreciate it. So I think with that, we can get started. Let me just make my slide a little. Okay. Okay, so basically I'm going to give, set the stage for the agenda here, and then I'm going to pass it off to Colin, who's going to give most of the demos for the webinar. So as I said today, the focus is going to be on data analysis, visualization, and exploration. We refer to this as DAVE, so we might use those terms interchangeably throughout the webinar, DAVE or DAVE tools, etc. Give you a few demos. We're going to show about project-level data visualization, survival analysis, visualization of mutation frequency. We're going to talk about creating custom cohorts, and then we're going to have a couple demos using our analysis tools, which are to create perform a custom cohort analysis and do set analysis. And then, as I said, we're going to leave a bunch of time for questions. Okay, so what are DAVE tools? I'm going to give you just a tiny overview of that, and Colin's going to give you most of the details. So the GDC provides user-friendly and interactive tools supporting gene and variant level analysis. What we are going to show you how to do is to visualize the most frequently mutated genes and mutations for a project. And just before I forget to mention, the visualizations that are currently live in the GDC are focused on those derived from whole exome sequencing data. So if you're wondering where we got these variants, that's where it's from. We're going to plot cases or patients with high impact mutated genes. We will perform survival analysis for cases with or without a mutated gene. We can visualize mutations and their frequency across protein coding regions. We will show you how to view the distribution of the number of cases affected by mutation across different projects. We will build cohorts and perform gene and variant level analysis on the cohort. There we go. Okay, so not to give too much away, but here are some of the plots that you're going to see, and we will explain in detail. On the left, we have some plots of the most frequently mutated genes in the survival curve. Zooming in on that survival plot, we have some lollipop plots on the lower left. We're going to show you more about those, and that shows you, where mutations are in terms of their protein domains and their frequency. And then we will show you a little bit about this oncogrid that, in this case, shows high impact mutations. So with that, I'm going to stop sharing my screen, and we'll roll here, and we'll give some demos.
Sure, yeah. So as Michael said, my name is Colin Reed, and I'm going to basically give an overview and do a lot of demos of these data visualization features here. So I'm going to share my screen. Let's see. So hopefully you should be able to see that. Yes. Okay, perfect. So as Michael introduced, there's a lot of visualization and exploration features available on the data portal. So I'm going to jump into some of the project level visualization, survival analysis, and then drill down a little bit further. And then as I go along, the demonstrations are going to get more complex as I go. So start easy and get a little more complex. So the first thing that I want to show is kind of an overview, the visualizations that give an overview of top mutations and top mutated genes in cancer patients. So basically, the place that you would want to go to see, for this example, I want to see the top mutated genes for lung cancer patients. I would go to the projects page, and then if I click on this, there's a few places to visualize this, but the projects page is a good place to see a high-level overview of all the different projects. So at the top of the screen, you can see a high-level overview of the top mutated cancer genes in selected projects. So I've selected no facets here, so basically it's visualizing all the top mutated for every single project. So as I said before, I want to look at lung cancer cases. So what I can do is, in some of these filters here, I can search for lung. If I click on that, you will see the visualizations dynamically change as I add these filters here. So when I click on that, I'll also click on TCGA. You can see that for lung cancer cases, TP53, KRAS, these are the top mutated genes for all of the cases that have that data available. And then you can kind of hover over these and see by a sub-project level which percentage of cases are affected by these mutations as well. So yeah, you can add additional filters here, but the next demo I'm going to kind of show is you can add a little more detail to these visualizations, and that can be done on the exploration. So the second demonstration I'm going to show is to dive a little bit more into this lung cancer cohort. So I will want to see kind of the survival of the lung cancer patient. So you can do this on the exploration page. So I will add that filter again here. Yep, lung, and I'll choose TCGA. And then if I click on the genes tab here, you will see essentially the same plot that was in the projects page, the distribution of most frequently mutated genes. And then, like I said, you have another few options here for visualizing this. You can add additional mutation filters. So in this case, I'm interested in maybe protein coding genes. I'd get a different visualization for that. So essentially, this demonstration, I wanted to show the survival plot on the right. And what this is showing is the survival of all of the cases that meet these filter criteria. So on the y-axis, you'll see survival rate. X-axis, you see a duration of time in years. And essentially what this graph is showing is the overall survival, or essentially the fraction of patients that are living after a certain time. Not all of the patients within the GDC have that survival data, so like the gene visualizations, it's only showing the cases that do indeed have follow-up data or additional data that contains survival data. And that's 1,006 cases for this. So this is the overall survival of this cohort. If you scroll to the bottom, you can also add, you can also compare the survival of two different groups based on a couple of different parameters. So the first one I'll show you is by gene. So you can compare different groups on whether or not they have a mutation on a gene. So if I just click on one of these boxes on the bottom right, you'll see them here. Let's see, I'll just click on this one. Clicking this button, what that does is it breaks the cohort into two different groups of patients. The first being those that are not mutated, and the second being those that are mutated. And you can see visually the difference in survival based on those two groups. And then you can click on another one if you'd like. So like I said, this is all interactive, and you can really easily visualize this data on the portal without actually having to. The other thing you can do is not only look at individual genes, but you can do the same thing with mutations. So if I click on the mutations tab, you see that same survival plot based on the cohort that I've selected. So if you're interested in maybe a specific mutation, you can click on that button again, and it's going to again break the cohort into two different groups with the mutation, and then one without that specific. And then the other thing that I did forget to mention is it does provide a log rank p-test value, so you can see statistically if there is a significant difference between the two groups. So yeah, so that's kind of the second demo I wanted to show was looking at the survival. This next demo, I wanted to show a visualization we put on here called the OncoGrid, and it's basically an advanced kind of heat map that shows some data across a few different variables. So I'm going to filter this down to make it a little more visible here. So I will just click on, for example, the TCGA LUAD cohort, and then maybe only those cases that have acinar cell neoplasms. I'll just click on that. And then what that does is it updates this OncoGrid kind of in real time. And then I will explain this in a little bit more detail here. So along the x-axis is a particular patient in the cohort. So each one of these columns represents a case. So as you can see, there's 22 cases at the top, so there should be 22 cases kind of going across here. And then along the y-axis, different top mutated genes in this cohort. So these would be the most frequently mutated genes. And then I'll go through one of these columns just to describe what's going on here. So let's look at one of these cases. I'll just look at this one kind of in the middle here. So this case has 18 total copy number variations, 14 mutations, and then each one of these cells in this heat map describes, it basically points out if a patient has a mutation on that gene or a CNV on that gene. So the dots represent mutations, and then each of the boxes represents, I can scroll to the top, either a copy number loss or a copy number gain for that. So it's a very high level visualization of patients on the x-axis, genes and mutations kind of sprinkled in to give an idea of what stratifies the cohort into more detail. Let's see. Yeah, I'll mention these things at the bottom. So these are some additional data. So it'll tell you whether each of these cases has clinical data and then what types of data are available if you wanted to look at these cases outside of the portal. So this is showing we've got clinical, biospecimen, copy number variation, transcriptome profiling, et cetera. So yeah, that's the OncoGrid. Again, it's a high-level visualization of what's going on for the cohort you've selected.
And then I'll just add, in terms of the OncoGrid discussion, as Colin mentioned, it's really for stratifying the data. And what you might see, is that certain patients, certain genes kind of cluster together or are related. Maybe there's a bunch of patients that have similar mutations or a bunch of genes that are found together or more frequently mutated together. And so this is really just to kind of, at a very high level, kind of look for patterns in the data.
And one more thing I'll mention here. So all of the figures that I've shown you so far, the survival as well as the OncoGrid, you can download the raw data from these, clicking some of these buttons at the top, or you can download the figure if you wanted a publication-ready figure or you wanted to share that with someone else. So the next thing I'm going to show is digging a little bit more in detail with the mutations that I had shown previously. So we've got a list of all the mutations in this cohort. So what you can do is, if you wanted to look at the mutations in the context of a location on a particular gene, we have some visualizations to show you that information. So what you can do for this cohort is I can choose one of these mutations. So that's one of the most frequently mutated genes here for KRAS, for example. I'll just click on that. And what that does is it jumps me to an actual page describing that mutation. There's a little bit of summary information. What's the DNA change, and then some external references you can click on to go to external databases to learn more about the mutation, and then the consequences. But the two plots that I want to highlight here are this cancer distribution plot. So for this particular mutation, this will show you the number of cases across the entire list of projects that are available. So you can see for this mutation, the project with the most number of cases affected is the TCGA I think it's a pancreatic cancer cohort. Pancreas. So there's a table down here. You can check that out as well. So yeah, this is just another way of visualizing the data. But the main, the meat of what I wanted to show you was this lollipop plot at the bottom. So for this mutation, like I said, maybe you wanted to visualize it in the context of the gene location. This plot essentially shows you on the y-axis the number of cases affected by a particular mutation. It's highlighting right now the mutation that I've chosen in red on the lollipop plot here. So that's the one that I chose. And then on the y-axis is the actual amino acid location on the gene transcript. So you can see particularly as I mentioned, it's a very frequently mutated gene on this protein, and this is what this plot is showing you: is there's a region of there's a bunch of cases with a particular mutation, and then there's a number of ways you can interact with this. So by default, it's showing the canonical transcript. You can choose a different transcript that is an ensemble ID, and it will change the plot dynamically. You can also kind of zoom in. So if you got like a huge gene you're looking at the bottom, you can maybe just select a particular region, and then it'll zoom in so you can get a little more granularity what's going on. So I can reset the plot. And then on the right, you can choose a number of other options. So if you wanted to only look at maybe the missense mutations, you can get rid of some of these here. Additionally, you can say I want to look at the data basically on an impact basis. It'll color all the mutations, whether or not they are high impact mutations or moderate or low based on VEP. So these are showing that this high impact mutation is actually, let me back up. So it's showing this most frequently mutated gene is categorized as a moderate impact mutation. So yeah, like I said before, you can download this as data or get it as an SVG or PNG image. You can use it for something else. So those are some of the visualizations that are available on the exploration page projects. But now I kind of want to jump into this custom set analysis capability of the GDC, because you can do a lot more powerful things with custom cohorts that you've created. So I'm going to jump back to the PowerPoint, and then there's only a few more slides, and then I'll jump back into some demos. So play from that slide. So with the GDC, you can do custom set analysis based on some case gene or mutation sets that are either created in the portal, or you can create outside of the portal and kind of use in itself. And then there's two basically analyses that you can do right now with this custom set analysis. You can do set operations, which lets you see unions and intersections of these different sets, as well as do a comparison of the individual cohorts based on some characteristics, such as the survival or diagnosis, vital status, etc. So I'm going to show you basically the procedure for going through creating a custom set in the GDC so you can do some of these analysis. So the first thing is to create the sets. How do you do that? You can do it either two ways. You can create custom lists, so you can upload TCGA barcodes, ensemble IDs, IDs, whatever you want into our upload feature, and then it will identify those and then create the cohorts themselves. Or you can use the existing filters, kind of like how I showed you before, where you can create a custom cohort in the portal and then use it for some of these analysis features. So after you've created the sets, you'll be able to see them in the portal. You can add additional sets, you can modify them, delete them, export them. And once you've got the sets in the portal, you can perform these analyses that I talked about. So you can do the survival analysis, you can do the set operations. I'm going to demonstrate some of that right now. I'll jump back to the portal. So the first thing I want to demonstrate is I wanted to do a cohort comparison of stage 2 and stage 3 kidney cancer cases and kind of compare what the different variables are between those two different sets. So the way that I would do that in the portal is by going to the exploration page, and then I can choose kidney, and then CGA. There's 943 cases for this cohort. So what you can do is we have a number of clinical and biospecimen fields that have been indexed into the GDC, and you can use those as filters to kind of filter out cases based on tumor stage, as I'm going to show in this example. So what I can do is add a case filter, and then you can see there's a number of different fields you can search by, but I know that I'm interested in tumor stage. So what that represents is the extent of the cancer in the body. So if I just click on this, you'll see a number of different options kind of show up here, but I know I'm interested in the stage 2 and stage 3 kidney cancer cases. So I will first create the stage 2 cohort, and I would just click on this button here, and then it filters the results on the right. So now you can see there's 111 cases that have stage 2, and they're basically the clinical metadata that's showing. Once I've created this cohort, there's a button on the right that says Save and Edit Case Set. So if I click on that, I can save as a new case set. And I'm going to, by default, choose the entire list of cases, and then it came up with the name stage 2 kidney. You can change it to be whatever you want. So I'll just click Save. So I've created my first set, and then for the second set, I kind of do the same thing. I'll just click on stage 3. And you can see that shows 196 have the stage 3 diagnosis, the tumor stage. And then I'll create that set as a new case set. I'll click Save. And then actually, let me go here. All right, so now that I've created the sets, the place that you can see and visualize the sets that you've created is this button at the top called Manage Sets. Clicking on that, I see the 2 sets that I've created. And then, like I said before, you can add additional sets. Like, for example, if you had a list of TCGA barcodes, or you wanted to look at specific genes, you would upload. You just click on this button and upload a list of those. But I've already created those. So the next thing that I will show is actually how to do an analysis with these sets that I've created. So if I click on the analysis button, you'll see a list. These are the 2 types that I had described before. So you have set operations and then cohort comparison. So the one that I wanted to do was a cohort comparison because I wanted to look at the survival differences between both of those. So if I just click Select, I can choose the 2 sets that I've created, and then I'll hit Run. And then you can see these are the 2 sets, the cohort S1 and S2 are stage 2 and stage 3. You can see the survival between the 2 sets. You can see information about different variables such as gender, age at diagnosis, things like that. And you can do this with any sets that you've. So the next demo I wanted to show, there's only a few more demos I've got, is to do a comparison of different sets. So I've already got these kidney cohorts already created. What I want to do is actually look at the differences between the genes that have been mutated between both sets, and I'll create 2 new cohorts to do that. So I'll just click on these buttons on the right, open these in new tabs, because I'm going to create basically 2 more cohorts of just the genes that are mutated. So I go back to the exploration page, but now I want to create gene sets. So the way that I can do that is I click on the Genes tab. So I've got a list of all the genes here. Instead of adding all 13,000 genes, I only want, for example, ones that have been tagged as being part of the cancer gene census. So I'll add that filter here. And that lowers this number to 415. And then here, this button's in kind of the same place here. You just click on this, save this as a new gene set. let's see, so this was, maybe I'll just call this set. And then I have that other tab open where I can essentially do the same thing. I'll just choose, is the gene part of the cancer gene census? I'll click on the genes tab. And then I save this as a new gene set. I think I call it set to something. There we go. So now I've got, if I go back to the Manage Sets page, the other set. That's strange. Interesting. Try that again. So let's see what it's got here. Stage 3, set 2. All right. These are 3, set 2. So let me save this as something different. Set 1. Oh, that's interesting.
Colin, it's saying that there's another set with the same name already exists.
Yeah, but it doesn't look like there is.
Might have to refresh it.
Try to refresh. It might be the 2 tabs.
Yeah. Let me try this again. So I already have the stage 2. Let me try stage 3. Cancer gene census. I'll click on the genes. And then I'll save this as a new gene set. I'll call it, I think the other one was set 1, 2, so I'll just say set 1. Click save. Oh, yeah, that's strange. That seems like it's a bug of some kind we're finding on a live demo here. What I can instead do is, I can pretend essentially I've got both of those sets here, but instead I'll just cases in the same way that I did, or I was planning to do with the genes. So what you would do is you can, instead of choosing the cohort comparison, you would go to set operations. You would select the 2 sets. So I'll just pretend these are the genes at this point. So I just click on these 2 sets of cases and hit run. And then what this does is it shows you a Venn diagram that essentially shows the overlap of sets that you've created. So if I had created the gene sets, what it would show me is what are the genes that are exclusively part of this cohort, what are the genes that are exclusively part of this cohort, and then an intersection of the 2. So if you wanted to see maybe an example of what that would look like outside of the portal, you could do an external analysis here. And I think one of the main uses of this feature that I think a lot of people find a lot of use in is finding cases that have data that are parts of 2 different experimental strategies. So what you could use this for is, let's say you're doing an analysis and you wanted to know, I want to find cases that have both RNA-seq data and full exome data, because you can't do your analysis without both sets of data. So what you could do is 2 sets of cases with 2 sets of files. Actually, I probably could demonstrate this right now. That might be a good idea. So what you could do is, if you were interested in doing an analysis with, so go to kidney, and I would choose, say I'm on the repository page right now, and let's say I wanted to find those kidney cancer patients with whole exome data and RNA-seq data. I would choose the 2 different experimental strategies here. So I will choose RNA-seq. Actually, did I choose cases? Yes. So I choose cases, and then I would save this as kidney RNA-seq as one set, and then I can choose whole exome as the other set, and then another set. So what I'm going to try to do, hopefully, yes, so it saved both of those. That's good. So what I was describing is you can use this set operations. So I will choose RNA-seq and whole exome. I'll just click on both of those and click run. What this is showing you is that intersection I was talking about. So overall, between these two groups of cases, there's 844 cases that have both whole exome data as well as RNA-seq data. So you could click on this maybe and say, I want to download the data for those intersecting cases, and then exclude the ones that for those. So that's one primary example that you could use this for. That's the two different types of analyses that you can do in the portal, and then I showed you some of the visualizations that we've got. So I wanted to kind of give you a little more advanced glimpse at what you can do with some of this outside of the portal. Because of the way we've designed the GDC, you can do a lot of these analysis in the portal. We've made the APIs public so you can fetch data and do an analysis outside of it and then kind of interoperate with the GDC. So for this last demonstration, I want to show you kind of an example of looking at survival based on gene expression data. So the gene expression data we have in the GDC, you can download it, do your own analysis, and then kind of in tandem look at some of that with the information that we have in the portal. So what I've done is I've actually created a Jupyter notebook where I've downloaded some of the gene expression data for a cohort into the notebook. And I'm not going to go into details of all of this analysis, but I just wanted to show you how easy it is to be able to interoperate with the GDC. So there is a tool that the GDC has called the RNA-seq tool to download this expression data. That's what I did. And then it really nicely formats it into this matrix so you can see all the genes as well as all the patients with gene expression data. But basically what I want to show in this example is I want to create a custom cohort of those with low expression of a particular gene and a cohort with high expression of a particular gene and use some of these visualization features that I've demonstrated here. So what I've done is in this notebook, I basically downloaded all the gene expression data for the liver cancer cohort for TCGA. And then I arbitrarily chose a gene KDM1A and broke the cohort into cases with high gene expression and low gene expression. And then I got the TCGA barcodes for those different cohorts. And then what I can do is actually use those TCGA barcodes, upload them to the GDC, and then see the survival of, compare the survival of those different cohorts here. So that's what I'll do now is I'll just copy this list of TCGA barcodes that I've kind of created here, and I'll create a new set. So if I go to Manage Sets, Upload Set, Case, I didn't show this feature, but this is how you would add a specific new case set. So I will call this liver high gene expression. What I can do is just copy the TCGA barcodes here. What it does is it tries to match the barcodes that you've provided with the metadata in the GDC. If it doesn't find a match, it does some fuzzy matching, and it will try its best to match these to a specific case. So it did. It found GDC cases, so I will just take this as one set. So here, high gene expression, and then I'll go back to my little notebook, and I have a variable called lower quartile. Let me delete some of this. There we go. So here's all the low expression cases. I'll copy those, and then create a new case set. We'll call this liver low expression. And then so it found 101 cases and submit. So now I've got these two cohorts, and then when I mean the high and low gene expression, I mean just the upper quartile and the lower quartile have high and low gene expression. for KDM1A. And then what I can do is I go back to the analysis and select those two cohorts of high and low expression, and then click Run. And then you can see again cohort comparison, but instead of using something that I created in the GDC portal, I uploaded cases to the portal, and then I can just visualize the data really easily here. You can see there's a statistical difference in survival based on the two. High and low, I didn't put the actual gene name in here, but it's high and low KDM1A gene expression. So yeah, and then the other thing that I wanted to mention is a lot of these visualizations that you're seeing on the portal are backed by our API. So backed meaning running using our API. So if you wanted to create these survival plots outside of the portal, or you wanted to create some of the things that you're seeing on the portal externally, we have that very well documented in our documentation. And I will actually just very briefly show you that here. If you go to documentation, and then you would click on API, and then I think it is so DAVE tools. So here are all the very detailed information about all the API endpoints query to get all of the data that you've just seen visualized here. So we have endpoints for genes, mutations, copy number variations, analyses here. And then, like I said, it's really well documented, and we've got a lot of different examples of how, for example, if you wanted to get information about a particular gene, or scroll all the way down to the bottom, you will be able to see, here we go, survival analysis. We have endpoints for doing survival analysis kind of available for the users. So I think those were all the major demos I wanted to show today. And then I think I could probably just to Michael to wrap things up and then go into questions. I think that would be good.
Sounds great. Thanks, Colin. That was a good demonstration of all the tools found in the exploration area, but then also how you can use the GDC to enable more advanced analyses, whether you're doing this in a Jupyter Notebook like you did, or in whatever sort of analysis software you're interested in, and some great examples of how to use the analysis. We will have to see about why I didn't save that one set. However, yeah. That is something we'll look into. So just a couple things to mention. You saw a lot of neat tools in the GDC for subsetting your data, but I want to mention that there's a lot cooler tools soon, actually, to the GDC. So we will have a new webinar when that happens, but basically we're going to improve the ability to select or to subset their cases. So different ways of making survival curves out of, between arbitrary cohorts, the ability to bin your numerical or discrete variables to select a cohort. Data-wise, we're releasing some updates to disease-specific staging systems, so that will also be nice to allow for some interesting comparisons. So please, everyone, come back to the GDC in a couple months-ish, I guess, and you'll see some of these new tools come out. They're going to be really neat. Okay, so don't have too much to wrap up. Yeah, just wanted to give these different resources for the GDC. Colin's shown you many of these, but please, feel free to take a look at the data portal. You don't need a login, as Colin showed, so you can do any of those things that Colin demonstrated, actually, without any login whatsoever. That's really only needed for kind of the raw BAM files and some of the variant. One important line to highlight there is the support email at the bottom or near the bottom. You feel free to ask us any sort of question about what you saw today or about anything else. And then there's one more slide. Yes, I also wanted to make sure people knew about our next webinar, which we will be diving in detail on the different bioinformatics pipelines that use to generate the data in the GDC. The data we spent pretty much, or most of the day with in the exploration section is whole exome sequencing open access variants. But we do actually have a lot of different pipelines. You saw a little bit of RNA data, but we have some whole genome sequencing data available. You know, for RNA-seq number variation, you saw a little bit of today, which is also indexed. But we'll dive into how we generate all those data, what sort of pipelines we use, et cetera. So I think that's all we wanted to say. So we kind of wanted to turn it over to questions at this point.
Hi, Michael. There's a question in the chat about seeing the genes that are listed for the number of mutations regardless of their significance. For example, TTN, which is frequently mutated from being a very large gene. Is there or will there be a way to assess significance based on gene length?
That is a good question, and one that we've talked a lot about at the GDC, actually, since it is something that people like to control for. Can you bring up one of those pages just so everybody knows what we're talking about?
Yep. So I think it was in reference to the exploration page when I just kidney, TCGA, and you go to genes, and yes, there's by default it's showing some of these really huge genes.
You're going to see Titan, yes. Titan, for all of the listeners, I think is the biggest gene. So what you're going to find is while it doesn't have a lot of mutations per length, it does have a lot of mutations, so it's going to show up on a lot of lists. We have explored different ways to control for gene length in this case, but each of those mechanisms has potential downsides for skewing or for obscuring. The way that we actually will resolve this is by default, the cancer gene census option is going to be selected when you go to this page. So if you select that, the TT. So that's kind of the long answer to a short question of how do we adjust for gene length, is that's actually a surprisingly complicated question, and there's ways to resolve it, but this is the way that, at least in the near term, that the GDC is going to resolve it, is by simply selecting cancer gene census.
The other thing that I was thinking too is gene length is actually available filter through the API, so there is a potentiality of just maybe putting in, I don't remember what the field is, I would have to look that up, but I believe you can just add a filter to this that would say, just show me genes that are less than 200 amino acids, but it's not a default option here, but it is possible, I think.
Yeah, so no, that's a good point. There's certainly, we can enable you to do those selections, on your own, but the GDC doesn't have one by default in terms of how that gene is being displayed. Are there other questions? I'm not seeing them, Sharon, but if you're seeing them, please feel free to read some of them.
Yeah, there's a question on making whole genome variants available.
Also a great question. So right now, there's a couple answers to that question. It's a question we get frequently, but if you want variants from whole genome sequencing, you can go to what's called the legacy archive in the GDC. So the legacy archive has all the data that precedes the GDC, but for TCGA, that will include all the data that was generated for TCGA, and this includes whole genome sequencing variants. However, we are definitely working towards releasing our own whole genome sequencing. So this is, our own alignment, which is different than what's done in the legacy. We align everything to HT38, and then we'll have our own variant calling pipeline. There's a couple whole genome sequencing alignment files now available for various projects. So we're starting to release those, and we are kind of in the late stages of implementing our own whole genome sequencing variant calling pipeline. So I do hope we can release those soon because I know our users are very interested in seeing them. But yeah, right now, Colin's showing you that you can find whole genome sequencing alignment files for a variety of projects. Not yet for TCGA, but those will be coming. And then hopefully soon for all the projects, we'll have the whole genome sequencing variants as well.
Michael, I have some more questions. Are variant annotations from ClinVar, OncoKB, Civic integrated in GDC? If not, are there any plans on integrating such annotations?
There are various annotations that are available. I don't know if you can click on a gene and show what is there. We are actually releasing Civic annotations next release, but right now we don't have ClinVar. Here's what's being added for our mutation annotations at the moment. And will you also go back to show what they can select for on the faceted search for mutations? And mutations. Just looking at the left panel to see the different types of information. Okay, so the answer is Civic is going to be added very soon. Actually, I don't think there's any immediate plans for ClinVar. I might not have the most up-to-date information on that, though. So I don't know, Sharon, if you have any more information on that.
For the most part, we're focusing on Civic in the next release, not ClinVar yet.
Yeah, that was my understanding as well. Thanks.
Okay, we have a couple more questions. Any plans on providing links to ClinGen allele registry?
I am going to have to admit that I don't know much about that. I'm going to write that down and learn more about it and talk to the bioinformatics team to have a more informed response to that. So if you want to ask, I would appreciate for that particular user if you would send an email to the support desk and give me some more information just so I can give you a more informed answer than that.
Okay, the next question is, can you run mutesig or the like and have an option of seeing that output?
Let's see, we do. I'm just trying to rack my brains. I think we are running mutesig, but I don't know if we have worked to make that information available yet to the community. It's something that we have kind of played around with here. So that is also a question I'm going to have to look into a bit more because I would like to know the answer to that too.
Okay, and another question is, if comparing gene mutations from different sources, what is the right way to do that?
Gene mutations. Oh, I think maybe the user is concerned about batch effects. That's kind of my read of that question. And so maybe just a little background on the GDC in that we do run the same pipelines, so we try to minimize batch effects to every extent possible. And that's one of the real virtues of doing analyses in the GDC in that pipelines are all identical across all of the projects that are released. We can't control for everything. There's certainly going to be changes to maybe how the samples were processed or something about the sequencing. So there could be, potentially some batch effects, although they should be minimized. So I think the short answer is you should be able to, as long as you're looking at the same variant caller. It's a great idea, of course, to look into any of the additional metadata that we might have for the biospecimen information to see if there's some other differences that might have led to that, such as whether it was FPE or what type of tissue that was derived from. Try and think of any other interesting metadata that might impact variants that are found. I think that would be where I would start in doing that. But that's certainly what we are really all working towards, is for researchers to be able to just run those comparisons without, for the most part, worrying about those additional details, since we are holding all of the constant. Yeah, feel free to, if you have like a specific question about different projects, the team is happy to look into that and see if there might be some other interesting variables to consider in doing analysis. Are there any other questions, Sharon?
No, I think that was it.
Okay, I do not see other questions either. So just a last reminder again to join us in about a month for our next webinar on GDC bioinformatics pipelines. And with that, thanks so much, everybody, for joining us. Here. Bye.